So today I want to talk about my favorite subject, DNA repair. The Photozyme MD products have fully embraced the importance of DNA in healthy skin and the need for fast and accurate DNA repair to preserve skin health and youthful appearance. So why is DNA repair so important? Well, first and foremost, DNA damage causes mutations in skin DNA, and this you're all familiar with. These mutations are central to the development of skin neoplasia. First, we're all aware of actinic keratoses. These are precancers, which contain all the mutations that are found in frank neoplasia. So they accumulate mutations, and then those are sorted out, and the drivers drive them on to develop usually squamous cell carcinoma. But then we have the category of non-malignant skin cancers, basal and squamous cell carcinoma, and those overwhelmingly contain mutations in oncogenes that have the signature of UV damage. So we can trace UV damage directly to the changes in the genetic code which lead to these cancers. And then finally, we have melanomas. Here, the link to sun exposure is clear, but the process is more complex. It probably involves the number of intense exposures, sunburns, rather than the accumulated dose. And that's because these intense exposures particularly burden the DNA repair process. But beyond mutations, I want to discuss the importance of DNA security in many other aspects of skin health, particularly skin aging. In particular, I want to introduce the concept that DNA is a sentinel for skin health. What do I mean? DNA is the most sensitive molecule in the skin to environmental attack, and it can alert the rest of the skin when damage has occurred. So in order to explain this a little bit better, let me take you down to the cell level and walk you through the first seconds and minutes, hours, days, and weeks after DNA damage. Now remember, DNA is in the cell nucleus. It's a double helix, it's twisted and torqued. It's coated with proteins that read the code and translate that information into cell function. And every so often, the DNA has to strip, unwind, and duplicate for cell division. Now, this DNA is under constant attack. Just living at 37 degrees, 98.6 Fahrenheit causes chemical reactions that damage DNA. And then on top of that, there's solar UV, which is the single biggest environmental cause of damage. The estimate is that a typical cell in the skin, every single cell in the skin, suffers about 10,000 lesions a day. Every cell has to deal with 10,000 attacks every day. So to better understand what happens as a result of DNA damage, let's walk through what happens after a sunburn. So first, there's the initial damage. DNA absorbs a UV photon, causing a photochemical reaction that binds together two adjacent DNA bases. And we call the most common one a cyclobutane pyrimidine dimer, a dimer because they're connected. This causes a kink in the helix, and it dislodges some of those bound proteins. In addition, free radicals, which are generated by oxidative reactions caused by UV, can also break the DNA, and this unwinds the torque that's in the helix. So within seconds, proteins that are specially designed to recognize these distortions now bind to the DNA. They're not bound until they see this kink or this break in the DNA. Some of these proteins which bind to the DNA are signaling proteins. They're called kinases, and they phosphorylate other proteins, which start a long chain reaction of signaling events. One kinase kinases another molecule, that kinase is another molecule, and you get this signaling reaction that tells the cell and all the cells surrounding that damaged cell that something's happened, I've been hit. Other proteins are then recruited to the site of the DNA repair protein, and they cover the wounded DNA and attract more DNA repair proteins to fix the damage. This all happens within seconds. Within minutes, transcription of the DNA is halted. The cells cannot use that portion of the damaged DNA to transcribe instructions. This happens in minutes. The signaling pathways continue to activate a chain reaction starting from these damage recognition kinases. And one of the targets of this chain reaction is the famous p53 protein. Now, this protein has been called the guardian of the genome, and that's because when it gets activated, it's able to activate a large number of DNA repair enzymes. But the p53 protein is a fascinating protein. It has many sites on it for phosphorylation, and so it acts sort of like a computer. If it's only phosphorylated a little bit, it only generates a small reaction. If it's phosphorylated substantially, it invokes the full complement of DNA repair reactions. But if it receives too many phosphorylation hits, then it signals cell self-destruction. It starts a program to destroy the cell, which is called apoptosis. So p53 acts as a computer to calculate how much damage has occurred. Now, in addition, there's a state that cells can enter, particularly aging cells, that's called senescence. And these cells are essentially cellular zombies. They're not alive, but they're not dead. They're not functional, but they're not apoptotic. They just sort of sit there and release signaling proteins that are damaged proteins. This is called the SASP, the senescence-associated secretory phenotype. And when you accumulate these senescent cells in skin, it gives rise to all the aging phenotypes. The skin isn't dead, but it doesn't respond the way youthful skin does. Now, within minutes of these, in addition to the p53 activation, there's a release of master cytokines, IL-1 and TNF-alpha. These are cytokines that then trigger the release of other cytokines. So you see this amplification, this cascade and amplification beginning within seconds, then minutes, and signals are sent out. These IL-1 and TNF-alpha that are released from the damaged cells cause a release of downstream inflammatory cytokines. They create an inflammasome that spits out these inflammatory signals, and some of these inflammatory signals recruit immune cells to the site of damage. So this all begins within minutes. Within hours, the signs of inflammation. We've now reached the end of these signaling cascades. The inflammatory cytokines have done their job, and now you start to see the physiological effect, the dilation of blood vessels, the recruitment of inflammatory cells. And by 24 hours, you see the physiological effect, a sunburn, which consists of inflammation, cell blood vessel dilation, and other inflammatory signs. If you have too much of these cytokine release, you have a cytokine storm, which we recognize as heat stroke. Now, not only is there an inflammatory response, but these signals within hours lead to tissue remodeling and protection. And here is where we generate all the signs of aging. First, we get release of MMP1, which is a metalloproteinase. It chews up the collagen. The intent is to remodel the wound, but in destroying the collagen, this leads to wrinkles. If you have repeated cycles of these release of these MMPs, you destroy the collagen, you don't have the supporting structure, and thus wrinkles develop. The same thing's true of elastases, which are released as a result of these inflammatory signals. They chop the elastin, and although the elastin isn't lost, it no longer has its function of providing some support to the skin. In addition, their melanocytes are given the signal to make pigment for skin protection. We recognize this as a tan, but often it's uneven, and you have signs of uneven pigmentation, which take a long time to resolve. So people ask me, and I'm sure they've asked you, how is DNA repair an anti-aging treatment? I thought it was just about cancer. And the answer is because DNA repair stops the signaling that leads to the release of these enzymes that cause wrinkles and hyperpigmentation. So DNA repair goes to work within hours after the initial damage. The human system involves a complex of dozens of proteins that have to find the damage, unwind the DNA, cut out the damaged strands, and then fill in with a patch. This takes time. After a sunburn, it takes 24 hours to remove half the damage. So you get a sunburn, the next day, you still have half the damage in your DNA. And the next day, you still have 25% of the damage in your DNA. So it takes time for the normal repair system to remove these damage. So clearly, there's an opportunity and a need to speed up the removal of damage so you can shut down these signals, not only prevent the mutations, but shut down these inflammatory signals. This is the opportunity to improve DNA repair for which the Photozyme MD products are designed. Now, within days, if these signals are not controlled, slowly the immune response peaks and it converts from what's called a Th1 to a Th2 response. The Th1 is the inflammatory response, but the Th2 response is an immunosuppressive response. The key cytokine here that's released after days is IL-10. And this suppresses the T cell response so that antigens encountered a few days after a sunburn do not trigger an immune reaction. And in fact, this immune suppression is key to the outgrowth of skin cancers. Skin cancers themselves can be removed by the immune system. But if you have immune suppression, then the immune system does not remove these skin cancers, nascent skin cancers, and they're able to grow out. So immune suppression is critical to the development of skin cancers, and these are triggered by DNA damage. Now, during these minutes and hours and days, it might be that the cell's time to divide comes up, but it can't divide because it has a damaged template. The cell may nevertheless still try to divide and replicate its template, but when it comes to a damaged part of the DNA, it makes a mistake in the copy, and that's when mutations arise. And they arise at these specific sites that are sensitive to UV damage. And so that's why we can trace a particular mutation to UV, because the mutations occur at sites where the photochemical reaction is most likely to occur. And that's why they're called signature mutations. Now, mutations can occur all along the DNA strand, and many of them might have no effect. But they do lead to drastic responses. First, they can lead to uncontrolled pigment production and persistent pigment production. And these lead to what we know as solar lentigines and post-inflammatory hyperpigmentation. But most importantly, the damage can occur in cancer genes, and the cell may then divide out of control. One example is a mutation in the p53 gene, which now is unable to control the other types of damage, and you get a proliferation of mutations, and those mutations can occur in genes that drive a replication. The guardian of the genome is essentially sidelined. So how can we stop all these reactions? The point is to speed up repair by adding enzymes that are able to help remove damage in the key moments after it occurs. And that's the purpose of the Photozyme MD products. They contain three important DNA repair enzymes. The first is called photolyase. This is derived from a plankton called Anacystis nigilans, and it repairs UV damage in a light-activated way. It binds to the DNA and absorbs light and uses that energy to split the pyrimidine dimer apart. Now, what type of light can be used to activate this enzyme? There's a broad spectrum of light that's able to activate the photolyase. UV, of course, can do it. Blue light is the most efficient, but visible light can also activate. So fluorescent indoor light can activate the photolyase. A rule of thumb is if you can see the skin, the photolyase can see the light. So unless you paint the skin or wear a piece of clothing, the photolyase is able to capture visible light and do its work. And this repair is much faster than the normal excision repair. The instant the photolyase absorbs visible light, it accomplishes its repair. It doesn't take 24 hours. The second enzyme that's in the product is the UV endonuclease from Micrococcus luteus. This is a cousin of the T4N5 enzyme, and it comes from a skin microbe. It repairs UV damage, it recognizes UV damage, but no light is needed. It's slower than the photolyase, but it doesn't require light. And overall, it speeds up the normal human repair. And the third enzyme is a glycosylase from a plant, Arabidopsis thaliana. And this is a glycosylase that's specific for oxidative damage. It can repair damage that's intrinsic, just arising from living in 37 degrees. It can repair oxidative damage caused by UV. And most importantly, it can also repair damage caused by air pollution. Now, one of the hot areas of research is the effects of air pollution on DNA and skin. One of the oxidizing characteristics of air pollution is called PM 2.5. These are particulate matter that's 2.5 microns, and they're little balls of carbon that are just coated with polyaromatic hydrocarbons. And once those bind to the skin, the polyaromatic hydrocarbons leach off. When they get to the DNA, they oxidize it. And this is a hot new area of research. So air pollution is an oxidizing agent, in addition to the well-known ozone present in some forms of air pollution, which is also an oxidizing agent. So the glycosylase is designed to repair damage from all these sources, the normal heat, UV, and air pollution. Now, these enzymes that are in the Photozyme MD products are real enzymes. There are dozens of publications on the molecular biology of these enzymes. There are dozens of clinical studies on these enzymes. Now, I've seen some other products, some crazy products that make crazy claims about DNA repair. Let me tell you, it's not so easy to make these enzymes work. So when you hear these claims, you have to ask yourself, do these other products support their claims with published data, or are they just hand-waving copiers? Now, the DNA repair enzymes are designed to complement, not replace, but complement sunscreens. Remember, the sunscreens stay on the surface, and the DNA repair enzymes work by penetrating into the living skin. The sunscreens must be used before sun exposure, but the DNA repair enzymes can be used before, during, or after sun exposure. This enhanced DNA repair can make up for times when the patients don't use a sunscreen, they forget to use a sunscreen, or they don't apply it correctly. So let's remind ourselves of the benefits of enhancing DNA repair. It's to keep the sentinel quiet. Not only does it lower the risk of cancer and precancers, but it reduces the signs of premature aging by stopping the signals that release enzymes that cause degradation of the skin structure and induce hyperpigmentation. And it preserves immune health and keeps our immune system from being suppressed. And I just want to remind everyone that it's really not too late to start. It's not just about the sun exposure when you were a child or a teenager. All these signals go on when you're exposed to the sun as an adult. So it's never too late to enhance DNA repair and stop the signals so that you can preserve your skin health.